Well, hello there and welcome back to our videos on the topic of cell division. So in this video, we'll be covering a very important part of this topic, which is looking at what are the stages of mitosis, as well as describing what happens during each stage of mitosis. Right, so these are the pages of your textbook which contain this information. Alright, so let's begin by recapping the cell cycle. So here's a diagram of the cell cycle. And if you remember, the cell cycle refers to a sequence of events from the time a cell is born until it completes dividing. So in this diagram here, the cell is born at about this time in the diagram, right? And then after that, it enters a phase known as interphase. Now, if you recall, interphase is a phase of growth, where the cell builds protoplasm and it replicates its DNA to prepare for cell division. Now, once the cell is ready, it will start to divide. And this starts with mitosis, which is a division of the nucleus. Now, mitosis has 4 stages, which we'll look at in a while. And then after dividing the nucleus, the cell will divide its cytoplasm in a process known as cytokinesis. So in the rest of this video, we're going to take a look at each one of these stages in greater detail. So let's begin with interphase. Now remember, interphase is not part of mitosis. It takes place before mitosis. And cells spend most of their time in interphase. Doing what? Well, they spend their time preparing for cell division. So they grow, they build more protoplasm, they make new organelles, and they replicate their DNA, they replicate their chromosome. So here's a diagram showing you what the cell looks like during interphase. Now during interphase, if you look into the nucleus, you will find chromatin threads. You will not find chromosomes because it is only during mitosis that the chromatin threads will condense to form chromosomes. Alright, and recall that chromatin is made up of DNA wrapped around proteins. So chromatin equals to DNA plus protein. Now during interphase, just before cell division takes place, the chromatin threads will replicate themselves. So the cell is getting ready to divide. And besides chromatin threads replicating, the pair of centrioles in animal cells will also duplicate themselves. So animal cells have this organelle called the centrioles. So normally they'll have a pair of them, one here, one here. And what will happen is that before cell division, the pair of centrioles will duplicate so that you get 2 pairs of centrioles in the animal cell. And we'll see in a while that these centrioles are important in helping the chromosomes to arrange themselves during mitosis. So after the interphase comes mitosis itself. And mitosis can be divided into 4 stages. Right? Prophase, metaphase, anaphase, and telophase. Some people like using the acronym PMET to remember the stages of mitosis. You can come up with some funny words to help you remember that. If anyone comes up with something good, please share with the class. So let's begin with prophase. Now early on in the prophase, some interesting things happen, as you can see already in the diagram here. Firstly, notice that the chromatin threads condense, and now we are able to see the chromosomes, the X-shaped chromosomes. So the chromosomes now appear, and they appear as a pair of sister chromatids attached at a centromere. Now why is it that they have this X-shaped appearance? Well, it's because these here are replicated chromosomes, right? Remember, X-shaped chromosomes are the replicated ones. So they are a pair of sister chromatids. There's a pair of sister chromatids, and we refer to this as still one chromosome. So there's 4 chromosomes, 4 replicated chromosomes in this nucleus. Right, next, you'll also notice that the 2 pairs of centrioles, one pair here, one pair here, will start to move towards opposite ends of the cell. Now, as the centrioles move towards opposite poles, they will also begin to radiate microtubules. So these protein structures called microtubules will start to form around the centrioles, and they form like star-shaped structures. So it looks a bit like a star, right? So they're called esters. Okay, next we look at the later part of prophase. So this is late prophase. Still prophase, but the later part. Now towards the later part of the prophase, you can see that the centrioles have already moved towards opposite poles of the cell. The esters are radiating from the centrioles, and some microtubules have started to form what we call the mitotic spindle. So this is the mitotic spindle. You can see some microtubules extending out, forming a spindle-like structure. So the mitotic spindle here is fully formed and is made out of microtubules or spindle fibers, which extend from one pole of the cell to the other. So if you look, if I just draw it out, you'll see microtubules actually extending out this way from one pole of the cell towards the other. And besides that, the nuclear envelope, as you can see here, and the nucleus break down. Now why is that? See, the nuclear envelope starts to break up and break down so that the spindle fibers in the mitotic spindle can start to interact with the chromosomes which are inside the nucleus. Okay, so after prophase comes metaphase. And here's a diagram of what a cell roughly looks like in metaphase. So here, right, you can very clearly see the mitotic spindle fully formed. Right, there are microtubules coming from one pole of the cell and extending to the other pole. And what you also see is that the nuclear envelope has completely broken down. It's gone. And the spindle fibers have interacted with the chromosomes and have attached to the centromere of the chromosomes. And the chromosomes have started to arrange themselves along the equator of the cell. Right, so putting that into words, right, the chromosomes, the pairs of sister chromatids, line themselves up along the equator of the spindle. Now this equator here is called, it can also be called the metaphase plate. So what we can say is that the pairs of sister chromatids line themselves up along the metaphase plate. And the centromere is attached to spindle fibers which come from both poles. So in fact, by this time, the pairs of sister chromatids are getting ready to be divided, right? So they've lined themselves up very nicely along the equator and they're ready to be pulled apart to opposite ends of the cell. And that's what happens during the anaphase. So over here you can see the anaphase. What happens is that the centromere of each pair of sister chromatids divides, right? So what was once here in the center has divided, and the sister chromatids separate, right? They were together just now, but they separate, and now they are pulled to opposite ends of the cell as the spindle fibers start to shorten. So these spindle fibers here attached to the centromere are shortening and pulling the sister chromatids apart to opposite ends of the cell. And once separated, each sister chromatid can now be called a daughter chromosome. So now, just now we had one replicated chromosome, right? But now we can say that we actually have two daughter chromosomes. So that once they're split, you can refer to each one of them as a chromosome. Right, so as the sister chromatids move to the opposite poles of the cell will also start to lengthen, and you'll notice that the nuclear envelopes will start to reform on both sides of the cell. And that's what happens during the telophase. So during the telophase, right, you can see that by now the mitotic spindle completely breaks down. So all the spindle fibers have broken down. Right, the nuclear envelope and the nucleus will reappear on both ends of the cell. And you notice that the chromosomes uncoil, so they uncoil back into chromatin threads. And now we're starting to see that, yeah, the nuclear division is more or less complete. Right? The nucleus has divided into two daughter nuclei. And as I mentioned, the cell continues to lengthen itself. So you start to see the cell lengthen itself, getting ready for the cytoplasm to be divided. Alright, so now that the nucleus has been divided, what remains is dividing the cytoplasm through a process called cytokinesis. And in animal cells, what happens is that a cleavage furrow is formed. So it's a little furrow is formed, and this furrow will deepen and deepen and deepen and deepen until it splits the cell into two daughter cells. Now at this point, I should mention that there are some differences between plants and animals when it comes to mitosis and cytokinesis. Right, the first difference is that in plants, there are no centrioles. Right, but in animals, there are centrioles. In plants, right, there's no cleavage of cytoplasm during cytokinesis. While in animals, there's a cleavage of the cytoplasm, the formation of that furrow, which deepens and splits the cell into two. Now, the reason why there's no cleavage of cytoplasm in plants is because plant cells have a cell wall. Right, so in order to split the cell into two, you don't really need to form a furrow here. All you need to do is form a new cell wall here, and it kind of splits the cell into two already. Right, so how does the plant cell form a new cell wall in between the two nuclei? Right, it does so by forming what is called a cell plate. Right, so what you will see is that a lot of fluid-filled vesicles will move actually from the Golgi apparatus. These fluid-filled vesicles will bud off from the Golgi apparatus, and they will fuse in the middle of the cell. All the vesicles will fuse to form a cell plate, and eventually this cell plate here will form the new cell wall that divides the two plant cells. So now you get two plant cells. Right, so a cell plate forms between the two daughter nuclei by the fusion of these fluid-filled vesicles. So let's end off with a quick checkpoint to check whether your understanding of mitosis is there. True or false? Individual chromosomes can be seen during interphase. The answer is false, right? You see chromatin threads, not individual chromosomes. Second, an error in the replication of DNA would affect successive future generations of cells. Now this is true, right? Because the error will be passed on to those cells. A pair of sister chromatids contains two identical copies of DNA. So are the sister chromatids identical? And the answer is true, right? They're identical, and they are joined together at the centromere. And final one, if one daughter cell contains 36 chromosomes, the other daughter cell will contain 18 chromosomes. And you know in mitosis that this is false, right? The two daughter cells should be genetically identical, contain the same number of chromosomes as the parent cell and one another. Alright, so there you have it. We've been through what happens in the interphase, we have been through what happens in each phase of mitosis, and we've been through what happens in cytokinesis. So you need to be able to identify diagrams, you need to be able to describe certain key events in each stage. So go through your textbook, go through the set of notes again, and I'll see you in class.